This is an overview of the digestive system. This is the alimentary canal or the gastrointestinal tract. And this is where we digest and absorb our food. Now, when it comes to the alimentary canal, we run from the mouth, the pharynx, the esophagus, stomach, small intestine, large intestine. And then we've got accessory organs that we're going to talk about along the way, such as teeth, tongue, gallbladder, salivary glands, liver, and pancreas. Now, the process of digestion, we view our gastrointestinal tract as a disassembly line. Nutrients become more available to the body in each step. Now, there are 6 essential activities, and we can go through these one at a time. First, we have ingestion. Ingestion is the act of taking food into the digestive tract. After that, we have propulsion, so swallowing and peristalsis. Peristalsis are wave-like contractions and relaxation of the muscles of the organ wall. And let me move my slide up. It looks something like this. So this slide here, or figure 23.3, shows peristalsis and segmentation. So these are the wave-like contractions squeezing in behind the bolus of food and pushing it down. I always compare peristalsis to squeezing a tube of toothpaste. Okay, so back to our gastrointestinal tract activities. We had ingestion, taking in the food, propulsion, swallowing, peristalsis. After that, we have mechanical digestion. Mechanical digestion consists of chewing and mixing of food with saliva and even churning the food. So we still see mechanical digestion taking place in the stomach. So those are my first 3. My second 3 are chemical digestion. Chemical digestion, this is the catabolic breakdown of food. Then after that, we have absorption. That's movement of nutrients from the GI tract to the blood or to the lymph, depends on what type of food, where it's going. And then we have defecation. Defecation is the elimination of indigestible solid wastes. Okay, so those are the 6 essential activities of the gastrointestinal tract. Okay, the external environment for the digestive process, we got to look at regulation of this. Okay, so we're outside of the GI tract itself. And so the way we control the digestive process is through mechanical and chemical stimulation. Of course, we know that if we take in food into our body, we see stretch receptors come into play, something pulling that organ larger. Of course, that's going to start the process. Or something like osmolarity, the change of the water, that's going to change it. But we've got these extrinsic control centers in our CNS. So from the outside, you got to think about the sight of food or the smell of food. That's the external environment for the digestive process. These are extrinsic factors. So in other words, things that aren't being placed into the GI tract. So that's the extrinsic control system. And then, of course, you've got an intrinsic control system. So those mechanoreceptors, those chemoreceptors, those all fall within the intrinsic control centers. By the way, those mechano and those chemoreceptors, they respond to like stretch, osmolarity. pH is a big one. Your GI tract will respond to pH. Simply the presence of what's called a substrate or food in the GI tract can start, or if it can also end digestion. Okay, so these things initiate reflexes. And that then in turn activates, or it can inhibit, the digestive glands. Keep in mind that we want to take that food, we want to mix it with saliva to form a bolus. After that, we want to get it mixing with digestive juices along the way. So anytime it's mixing and anytime it's inside this GI tract, we call that the lumen. So let's jump up here a couple pictures. There we go. So here is figure 23.6. This is the lumen of the digestive tract. So that's where you find the contents of your food. I like this picture. I think it does a good job. Okay. Prior to this is figure 23.4. This just simply shows, there's the sight, smell, and taste of food on your CNS and what that does. And then, of course, the chemoreceptors also sending the messages to your brain to get your GI tract going. This shows the peritoneum and the peritoneal cavity. The peritoneum and the peritoneal cavity are the ones that house your gastrointestinal organs. Around the outside of your peritoneum, you've got a serosa membrane. Don't forget, when it comes to any of our membranes that are associating with our organs, we're going to have a visceral membrane, which actually covers the surface of most of the digestive organs. And then we're going to have a parietal. The parietal are the ones that are out here on the body wall. You can see there's the parietal peritoneum. And then up here touching the organs themselves would be the visceral peritoneum. There it is, shown right there. Okay, so the peritoneal cavity is the space between those two layers, and like what we saw with the heart, most of the time this is a fluid-filled cavity. It's there to lubricate the digestive organs, allow them to slide past one another so that we don't end up rubbing off cells and damaging the organs from the outside. Okay, I showed you this picture just a little bit ago, this figure 23.6. This is what we say is the histology of the alimentary canal. So in other words, from the esophagus to the anal canal, the walls of the GI tract have the same 4 layers or tunics, okay? From the lumen, we're gonna start here in the middle. I showed you the lumen earlier, that's the opening. The first one we go into on the way out, we have the mucosa. So anytime we have a mucosa, we know that we're going to have goblet cells are going to be lining this. So we have different glands of the mucosa. But anyway, this is my mucosa laying up here. And you can see that we've got a gland in the mucosa. As we move out from the mucosa, then we have the submucosa, which is my next layer. Oh, let's go back to the mucosa real quick. Let's talk about the functions real quick. The functions of the mucosa is to secrete mucus. Obviously, that's going to put moisture into the food. It's going to help it move fast. You get the idea. Mucosa is responsible for the absorption of the end products of digestion. And then it's also there to protect against the infectious disease. Now, my picture here is crazy, isn't it, with detail? You don't have to know all this detail, but the mucosa itself actually has a lining epithelium, and then we have a lamina propria, and then we have a muscularis of the mucosa. It's got its own muscle layer. Again, if you know that this is the mucosa layer, you're good to go for me. As far as the cells go in the mucosa, simple columnar epithelium with goblet cells. I think when we looked at tissues and we looked at goblet cells, I think most of the time we were looking at pseudostratified, but these are pseudostratified columnar. In this case here, these are just simple columnar layers, simple columnar epithelium with the goblet cells. In that mucus, besides mucus itself, obviously a high content of water, but you also end up with some enzymes secreting cells, and then also some hormone secreting cells. And we can get more into that as we move along. Okay, let's move out then to the submucosa right here. Next layer out, kind of thick. This is a dense connective tissue. It contains elastic fibers. Remember, once we put food in the lumen, this whole thing has to be able to stretch. So we do see elastic fibers in the submucosa. Other things we're going to find there, blood and lymph vessels, lymph nodes, and then nerves. That's the big thing, okay? But as far as functioning going, it's a protective layer because it is just, it's just dense connective tissue. Next layer out, we have the muscularis. Now, the muscularis is the one that's responsible for segmentation and peristalsis. So we'll spend more time on the muscularis because you can see from my pictures, sorry, I keep seeing something on my face. From my picture down here, we have a couple layers. We got a longitudinal layer and we have a circularis layer. That circularis is going to wrap around the whole tube as you go down. The muscularis is going to run longitudinal, so it's going to run along the walls. Now, you always find those 2 layers. When you get to the stomach, we pick up a third. We pick up an oblique layer, and we'll look at that when we get to the stomach, but we're not ready for that yet. But just know, in the muscularis externa, this is the muscle layer of your alimentary canal. Okay, my last or my outermost layer is a serosa, and it's a protective visceral peritoneum. This replaced by a fibrous adventia in the esophagus, but it's a connective tissue layer on the outside. It's funny because your retroperitoneal organs, so behind the peritoneum cavity, those tissues have both an adventia and a serosa. But anyway, here's my serosa. You can see my cells, epithelium and connective tissue. So, okay. So I think we're ready then to start with the anatomy of the mouth or the oral cavity. We'll get through this real quick. Oral cavity is bound by the lips, the cheeks, the palate, and the tongue. That's what makes up the walls on this. We have an oral orifice. That's an opening to our mouth. Anyway, and then our oral cavity is actually continuous with the oral pharynx. Remember that the oral pharynx was one of the three pharynxes that we had before in the respiratory system. Started with the nasal pharynx, then it went oral, then it went laryngopharynx. But anyway, so when it comes to the oral cavity, it goes straight into the oral pharynx. Now, the mouth has to withstand abrasion. So the tissue you're going to find there is stratified squamous epithelium. This is the same tissue that we found in our skin, because when you take a bite of that McDonald's hamburger, those cells are going to go with the hamburger. And if you have too much rubbing in there, if these weren't stratified squamous, we'd rub them off and lose a lot of cells. But these are layered, and they're also keratinized, so they've got another protein embedded with them, so it gives them just a little more strength to it. So, matter of fact, along with the tissues in there, your gums, your hard palate, and the dorsum, the top of your tongue, those are all slightly keratinized. So you've got that protein running throughout. Okay. Now, we'll stay here. You have a core of skeletal muscles. When we first started looking at muscles around the outside, we have the orbicularis oris. I asked you to learn that one. And then you have the buccinators up here that help you with the chew. Okay, the vestibule are bound by the lips and the cheeks externally and the teeth and gums. So in other words, when you puff out your lips, you're increasing the vestibule. The vestibule is different from the oral cavity proper. The oral cavity proper is the area that lies within your teeth and gums. So there is a difference between those two. So please make sure that you know the difference. Take a look at my picture up here. You can see that we've got what are called frenulum. Up top here, I've got a superior labial frenulum, and down here I've got an inferior labial frenulum. Frenulum are those flaps of skin that helps keep things attached. So we also have a frenulum on the bottom of our tongue to tie our tongue to the floor of our mouth. That one's the lingual frenulum. Let's see, up top here, we've got the hard palate, and you know as we move back, it becomes the soft palate because we lose the palatine bone. You can look at a skeleton on that and see that the palatine bone runs a short distance and then it ends. And so that's why you go from a hard palate to a soft palate. The hard palate itself assists in chewing, and it's slightly corrugated on either side of the midridge, just to hold food in place. If you've ever looked inside a cat's mouth, they've got big ridges inside of theirs, and that's to keep everything on the inside. That's important to them. And so, but they're slightly different. The tongue itself, this is going to occupy the floor of the mouth and fill the oral cavity when your mouth's closed. Take a look at this. When I saw that real head cut in half that time, I was amazed by how thick our tongue is. Functions of the tongue consists of gripping and repositioning food during chewing, mixing food with saliva to form that bolus. I think I've used that term a couple times. A bolus is food plus saliva mixed. Your tongue also initiates swallowing. So when you start the swallowing process, it's the tongue that does that. And then also speech is a big one. Now, you've got intrinsic muscles, so muscles on the inside that help change the shape of the tongue. And then you've got extrinsic muscles, and they alter its position, like sticking it out, things like that. I've already showed you the lingual frenulum on the bottom, so I don't need to cover that again. Now, the superior surface, you have three types of papillae. Here's what a papillae looks like. Looks like that. So here's another one. You have a filiform. Here's what filiforms. So they're kind of pointed on top, aren't they? And you probably guessed this is going to give your tongue that roughness, make it feel just not smooth. Cats feel a lot like sandpaper, and it's because of those pointed parts of the papillae. That was good. Secondly, we've got fungiform papillae, and those are scattered widely over the tongue, and they give your tongue the reddish color. That's where it gets it. And then back here at the back, you got these big ones. These are the circumvallates. They're the monsters. And they make this V-shaped row in the back of the tongue. Now, when it comes to salivary glands, your salivary glands are not on the surface of your tongue. Take a look. I'm showing you a papillae here. See how the salivary glands are down along the side? So that's why food has to mix with saliva, so then it can get down inside of these crevices on the side of the papillae. That's where we actually see the salivary glands come into play. I get the wrong picture. Anyway, your liquid food particles into these, they come into contact with they're like a hair cell, and anyway, that takes the whole thing, that moves this whole action potential to the brain. But take a look at our tongue over here. See that we've got sweet on the tip of the tongue, we've got salty on the outside, so we got sour back in the back on the sides, and then bitter at the very back. We've actually added a fifth one. It's called umami. And this is the metallic flavor. I can't remember which amino acid it comes from, but it's one of the amino acids. And that's how we get like the flavor from beef. Beef is, you don't have the bitter, salty, or sweet. It's a mix of those, but it's also a metallic flavor. So if you're dealing with something that has a metallic flavor, it's because of that other taste bud. Okay, enough of my taste buds. Yeah. Salivary glands, real quick, produce and secrete saliva. They cleanse the mouth. They moisten, dissolve the food chemicals. They aid in the bolus formation. And then they contain enzymes. This is the important one. They contain enzymes that help break down starch. That enzyme is called salivary amylase. That one's important. I absolutely think you should know that salivary amylase helps the starch process. So saliva itself is going to be 97 to 99.5% water. It's slightly acidic, and of course it's going to contain the electrolytes that we've seen all along: sodium, potassium, chlorine, phosphates, bicarbonate, that sort of thing. There's other proteins in your saliva, lysozymes, defensins, immunoglobulins, and then you also find metabolic waste. Urea and uric acid, like what you find in urine, that's also found in saliva. Okay. When it comes to the control of the salivary glands, you got to remember these things are here to keep the mouth moist and to help with food, to process food. But you've got strong sympathetic stimulations, and that's it, to inhibit your salivation, and that results in dry mouth. So it's those extrinsic salivary gland stimulus that actually get your salivary glands to respond. So ingested food stimulates those chemoreceptors and pressure receptors. The thought of food, that also gets the saliva rolling. If you've had psychology, you'll learn all about Pavlov's dog. And just in his case, Pavlov would ring a bell and give the dog food, and ring a bell and give the dog food, and ring a bell and give the dog food. Eventually, he would ring the bell and not give the dog food, but that would cause the dog to salivate. Good to know, right? Okay. Last thing on the salivary glands, and then we're going to keep going here. The three that you need to know: the parotid gland is up here. This is on the outside. When you dissect your fetal pigs, I always make my students find their parotid glands first because gland tissue looks very different. Out to the outside here, we've got the submandibular, and then down the middle, we've got the sublingual gland. Those are the big three that I think everyone should know. Okay, teeth, real quick. Let's end with teeth and we'll be finished with this. When it comes to teeth, you've got primary and permanent teeth. We also call these dententions. Your permanent ones have all formed by the age of 21. Your primary, you only have 20 of these things, and they start erupting or coming into your mouth from 6 to 24 months. And then your permanent ones, those come in basically from ages 6 to 12, but all the way up to 21 years of age. And we'll talk about those here in just a second. Usually you have 32 permanent teeth though. But take a look at the deciduous teeth up here. You've got two incisors. Incisors are the chisel-shaped ones. They're good for nipping. I always think about eating a carrot and you bite it off with your incisors. And you've got 2 of those. Now, when I say 2 of those, I'm talking about just on half of your upper, your lower. So overall, you've got 2, 4, 6. You've got 8 of them total. But for the sake of this discussion, know that I'm just talking about a line right down the middle here, and I'm talking about these 2 right here. Okay, so 2 of those. After that, we go around. We have a canine. A canine's kind of sharp. It's there for piercing, so it tears through things. I don't have a good example for you. We'll work on it. Maybe tearing, yeah, thank you. Maybe tearing chicken off the chicken bone. And that was Max sharing in my lecture. The next, we have the molars. We've got 2 molars in this case. We've got the first molars, and those are up between 10 and 15 months. And then the second molar about 2 years of age. So if we look at dental pattern, again, we're looking at 1/4 of the mouth. So the dental pattern here would be 2, 1, 2. And that's a deciduous dental pattern, or we call these your baby teeth dental pattern. Okay, let's move up to your permanent teeth. Here's your permanent teeth. Once again, 2 incisors right here on front, chisel shaped, 1, 2. Next, you have a canine, so you only have 1 of those, so 2, 1. And then you have premolars. Premolars are bicuspid. Bicuspid means they've got 2 surfaces. And then after that, so you've got 2 bicuspids, and then you've got 3 molars, and those have multiple surfaces on them. So your dental pattern becomes 2, 1, 2, 3. So for the grand total of 32 teeth in your mouth. Again, teeth are classified according to their shape and their function. Incisors are the chisel shape for nipping and cutting. Canines, fang-like teeth for tearing or piercing. Premolars, bicuspids, and molars both have that broad crown and rounded tips, best suited for grinding and crushing. During chewing, upper and lower molars lock together, generating crush force. I knew it was in there. So here's that dental pattern that I was telling you about, the 2, 1, 2, 3. You can take a look at that. Spend some time on it. The structure of the tooth. Oh my goodness, this is getting long, isn't it? I apologize now. When it comes to the structure of the tooth, some things you need to know. Your tooth is buried down into the bone down here. There's 3 things that keep your tooth into the bone. First of all, you've got the gums, which are known as gingiva from here on out. There's your gingiva. If you have gingivitis, that's inflammation of the gums. After that, you've got cementum. Cementum is a chemical that's actually produced by the cells surrounding your tooth, so that's like a glue. And then finally, we have the periodontal ligament. Now, for those of you who like Disney movies, if you've seen the movie Finding Nemo and know all your characters, at one point, the pelican, and what's the pelican's name? Nigel, good. Hopefully you said that yourself. I was waiting for Max to answer, he didn't answer. Nigel comes flying into the dentist's office, hits the window. At that point, the dentist was working on someone and pulls a tooth out. So the dentist makes some comment about pulling the tooth out, goes over and opens the window. And then Nigel sneaks in, and the starfish, Peach, says, "Nigel, you just missed an extraction." And Nigel makes the comment, "Did he loosen the periodontal ligament?" Okay, for once, Disney got it right, or Hollywood got it right. You would loosen the periodontal ligament before you extracted the tooth, because the gingiva, the cementum, and the periodontal ligament are the 3 things that are holding your tooth into your skull.
I was hoping you'd talk about Darla.
I don't like Darla. She's evil. Okay, so now let's talk about the rest of the tooth. So we've already covered everything down here, haven't we? Up here, we've got enamel. Enamel is made of hydroxyapatite. That's the same crystalline structure you find in bone, but remember that enamel is not bone. We know that enamel is the hardest substance in your body because, I'm sorry to say, after the fire, the only thing that's left over are teeth. We have to identify people by their dental records. Harsh, I know, but that's just the way it is. So enamel is harder than bone. Underneath the enamel, we have the dentine and the dental tubules. That's where you're going to find living cells, because remember, the tooth is an organ. And then underneath that, you find the pulp cavity, and then the pulp cavity that contains blood vessels and nerves. So, and then here's the root canal going down. If you've ever had, first of all, let's start with a cavity or a carie. That means the enamel has decay in it. You're breaking through the enamel. If it gets bad, it actually goes into the dentine. If it gets really bad, it goes all the way into the pulp cavity. You can not have, you may not have a cavity where you can still end up with an infection in your pulp cavity. I had that. I had to have a root canal. In the root canal, what had to happen was they drilled a hole down through the middle here, through my enamel, through my dentine, opened up the pulp cavity. They then cleaned all this mess out, all the way down, clear to the bone, taking out all of the blood vessels and all the nerves, everything that was there. They packed this tooth with an inert substance. They look like little plastic rods to me, but they eventually fill in. And then they just fill this whole thing back in. Sounds easy, doesn't it? It wasn't fun. I wouldn't recommend going for that. If it happens though, you'll be fine. I've had 2 of them now. One of them hurt really bad. The other was a piece of cake. Okay, what else? I think that is it for this video. I apologize. This was a long one, guys. So I'm going to go ahead and stop us now, and we'll move on later.